Good afternoon, and welcome to the Wednesday afternoon lecture series. I'm Andrew Griffith. I'm from the National Institute on Deafness and Other Communication Disorders. And today's talk is important because understanding voice, speech, and language and their associated disorders is critical for human patients, because impairment of these communication modalities has devastating effects on these patients, and these disorders include things like stroke, stuttering, dyslexia, and many others. So identifying animal models for such a trait has been somewhat of a challenge, and but songbirds have proven to be a useful model for aspects of vocal learning and production. And today's speaker, Dr. Eric Jarvis, is a pioneer in this field who is best known for his discovery of brain pathways and genes involved in learned vocal communication in songbirds. He's published over 60 scholarly articles, including a series of seminal studies in the late 1990s with Dr. Fernando Nottebohm. He's also well known for his personal and professional journey toward a career in research. He was born and grew up in Harlem, New York, where he attended and majored in dance at the New York Public High School for the Performing Arts. He was offered dance scholarships with the Joffrey Ballet and with the Alvin Ailey Dance School, but decided instead to attend Hunter College, where he received a bachelor's degree in mathematics and biology. He then pursued graduate and postgraduate fellowship training at Rockefeller, where he earned his PhD in molecular neurobiology, and began his lifelong work in vocal learning in songbirds with Dr. Nottebohm. In 1998, he joined Duke University in the Department of Neurobiology, where he's risen through the faculty ranks to a tenured position, as well as many secondary appointments. He's received dozens of awards and wide recognition, and is the source of CV envy for me. In 2002, he received the Alan T. Waterman Award, NSF's highest award for young investigators, given annually to one scientist or engineer who under the age of 35 made a significant discovery in science. And just a few of the other awards: in 2005, he received the NIH Director's Pioneer Award, and in 2008, he became a Howard Hughes Medical Institute investigator, and in 2012, he'll deliver the Wednesday afternoon lecture series. So welcome to Dr. Jarvis.
Thank you for that introduction. All right. So, yeah, I've been primed to say that this is one of the big lectures here, and so I hope not to disappoint. And I'm going to try to keep it general, but also really encourage questions towards the end, or if there's something that you don't really understand in the middle, I'll go ahead and answer it. So, my interest, yes, is in understanding brain mechanisms of complex behavioral traits. And the particular trait that I studied most is vocal learning, because it's considered one of the critical behavioral substrates for spoken language. And when I began this project, as you can see in this title, it lists 3 different species. The assumption was that you have humans who are vocal learners, and we use that behavior to produce and imitate our speech that I'm doing now. And songbirds, and parrots as well, who tend to be model species for this trait. At least that's the animal model that gets closest to what we can say of anything that's like speech. And then mice, who are considered non-vocal learners and can't be used for a disability. And so that's where I was beginning. And I'm going to talk about a project to really address that question: is that really true? And that work, as in most labs, is not just done by one person, myself, but done by multiple people. Where's the pointer? Is this it? Great. It was actually done by 2 people in my lab: Gustavo Arriaga, who now graduated and did a short postdoc in my lab, and will be starting a new postdoc soon, and an undergraduate student, Eric Zoom. And they really did a tour de force project over a number of years that I'm going to tell you about. So to give you some background here, let's ask, what is vocal learning and who are vocal learners? Vocal learning is the ability to imitate sounds that you hear. Some species can do it prolifically, like humans. Some species are very limited, like the zebrafinch, the songbird that we studied in the laboratory. Others, like a starling, can imitate thousands of sounds. And when vocal learning is present, what we've seen amongst the mammalian tree or the bird family tree is that it's relatively sparse. So here's one view of a mammal family tree, and regardless of the view that you look at, you'll see that those that are vocal learners, I highlight in red here, elephants, dolphins, and bats, whales as well, and amongst primates, only humans, not other primates. It's sparsely distributed amongst the mammalian family tree. The same thing for birds. So we have roughly 28 orders of birds here, and we have hummingbirds and parrots and songbirds that are the vocal learners. Now this is different from auditory learning, which is the ability to process novel sounds and learn something new about them. And you need auditory learning for vocal learning, but just because you have auditory learning doesn't mean you automatically have vocal learning. So it's been argued that this ability of vocal learning has evolved independently also in birds. One possibility, though, is that there's a new view of the avian family tree with some 16 genetic markers, has argued that pulled parrots close relative to songbirds, with the possibility leading to that maybe there was two independent gains of vocal learning, once in the hummingbirds and then once in the ancestor of parrots and songbirds, and then lost in subassine songbirds. And this would be the equivalent of humans and chimpanzees having a common ancestor with vocal imitation, and chimpanzees losing that ability and humans maintaining it. So whatever how this evolved, it's fascinating. But it's, all along it's been assumed that rodents do not have this ability. But once a species has it, it seems to come along with a package of traits. And that package, I've listed in several bullet points here, is that we depend upon auditory feedback to actually practice and develop our learned vocalizations. We go through critical periods. And when I say we, I'm talking about vocal learners generally, not just humans. We go through critical periods where we learn how to imitate vocalizations best at an earlier stage in life than after puberty. That's why it's easy to learn different languages before puberty. We culturally transmit vocal repertoires from one generation to the next, and we have some form of syntax to various degrees. Some produce more complex, some produce simple syntax. And disorders of vocal communication are also shared amongst vocal learners. One of them is deaf-induced vocal disorders. So when we become deaf, or when a songbird becomes deaf, our vocalizations eventually deteriorate if we don't have some type of therapy. The speech becomes muddled. And that is because we need to hear ourselves in order to maintain the learned vocalizations. Whereas non-vocal learning species, when they become deaf, the vocalizations remain intact due to innate. And we have aphasias of speech through brain damage, and for songbirds we don't call them aphasias, but you can damage the brain and affect the learned sounds. And speech sound disorders, we haven't found anything like that in songbirds yet, but there are people out there searching for that, including things related to autism. So that's the behavior. What about the brains? Well, we actually know more about the brains for these pathways in birds than we do in mammals, because we can do a lot of experimental work with them. And my work and others have shown that here's the family tree of birds, and here is semi-3D reconstructions of their brain anatomy for vocal communication pathways. Highlighted in blue here are brain pathways that are involved in processing the sounds that an animal hears, in this case these birds. And you can find this auditory pathway. I don't show the connectivity here, but necessarily you can find it in all species of birds, and you can find it in many other vertebrates in the forebrain. And it's thought to be involved in the processing of not only of sounds, but also in learning information about auditory signals. So it's been argued that this auditory pathway found in the vocal learning species, the songbirds, parrots, and hummingbirds, was inherited from a common ancestor. However, only in the vocal learning species, I've highlighted in red and yellow here, are brain regions that are responsible for acquiring, that is learning the vocalizations in this red labeled pathway, and producing those learned vocalizations in this yellow labeled pathway here. When it's found in these vocal learning species, what's interesting is that we find exactly 7 brain regions in all 3 of them. not 5 in one or 3 in the other. They're not in identical locations, but they have very similar connectivity. So it seems like once a species evolves this ability, it evolves it in a similar way. And when we published this at the time, several religious groups did contact us and said, this helps prove the existence of God, because how could you get such a similar pathway multiple times in the last 65 million years? And we didn't have an answer to that. Maybe there was a common ancestor and there's mass extinction of vocal learning, or there really are 3 independent gains. And, excuse me, I'm going to have to just do it from here. We didn't have an answer to that, but looking at the human neurobiology for vocal communication, learned vocal communication, come up with a similar scenario, that I've argued that we have a Wernicke's-like area that's homologous to these auditory forebrain areas in the birds, because you can't find a vertebrate group without them, that involved in processing species-specific or heterospecific sounds like, come here, boy, fetch the newspaper to your dog, all right? Or, and that we have the strip of cortex, parts of the striatum, thalamus, and the face motor cortex that's involved in learning and producing learned speech. For speech, I'm basically going to say spoken language. And that so far, these brain pathways I've color-coded in red and in yellow here cannot be found in non-vocal learning mammalian species. So we can argue a similar scenario here, that there was an independent gain. I don't think humans and songbirds and parrots shared a common ancestor and all the other species lost it. That is this pathway in the trait. So our goal was to test some hypotheses about genes that form these circuits that we're looking for, and to then put it, a crazy idea was to try to transfect those genes basically into the mouse genome and try to induce a vocal learning pathway. And in order to do that, we needed to decide or needed to know more information about the vocal system of mice. And I and many others had written in our reviews that mice are non-vocal learners. But actually, when you look at the literature, no one's ever tested that. It's been assumed. And then Tim Hawley from Washington University published a paper a number of years ago, some of you may have heard about, where he showed that mice have these ultrasonic vocalizations. That was already known, but they have characteristic features like songbird songs. And what you see here is a sonogram of the sound, time on the x-axis, the frequency of the sound on the y-axis here, and each one of these structures here is basically a single syllable morphing from one form into the other. And this is in the ultrasonic range, meaning out of our hearing range, 50 to 100 kilohertz. We don't really hear that well above 14 kilohertz. And so this is now pitched down to the human hearing range, and this is what it sounds like. I'm going to turn on the sound here. Now, when I play that, some people think they're hearing some kind of songbird. But if you pitch it down even, slow it down, this is what it sounds like. Here. All right. So, this is not a simple type of sound. It has some structure to it. It's a whistle-like sound. And they even found that different individuals here are 3 different individuals where they classified the syllables into 3 broad categories, are producing different proportions of their syllable repertoire amongst these categories. So in other words, there are individual differences, which is what you would expect in a vocal learner. So this led to a flurry of media and so forth suggesting that mice might be vocal learners, but this doesn't prove or one way or the other. It just says like they have a vocal behavior that has some features similar to songbirds, but not, that doesn't mean they're actually really vocal learners. For example, there are some songbird species, like this Oscine songbirds, that produce a song, but it's an innate song. So just because it's song doesn't mean that it's learned. So what did we do? We went to test out whether or not mice have this package of traits that you find in humans and songbirds and parrots and other species that have been tested. And we examined 3 brain traits, whether or not they have forebrain areas involved in the, or at least active in the production of vocalizations you find in vocal learners, but not in non-learners. When I say non-learners, I'm referring to here experiments done in non-human primates or pigeons or chickens, some in guinea pigs. One that's talked about a lot is that there is a direct forebrain projection to the brainstem motor neurons that control vocal behavior in humans and parrots and songbirds, but hasn't been found in non-human primates. Requiring the cortex to produce those vocalizations, the behavior itself, I mentioned auditory feedback and actual vocal imitation, and more recently, experiments that we and others have been conducting to identify genes that are associated with the evolution of speech or song in songbirds. So these two students, myself and a few others, we actually marched through these one by one, and we took a bottom-up approach, starting with the brain as opposed to the behavior. And we used our knowledge of the songbird system and of the human neurobiology for speech to formulate our experiments on mice. And so the first one is shown here. Work that I did actually as a postdoc shown that when songbirds produce their learned song, what happens, that behavior is associated with neural firing in the brain, which causes increased mRNA expression, shown in white here, of certain genes that are responsive to neural activity, called immediate early genes. And what you're seeing here is a sagittal section, thin slice, through a canary brain. The red staining is the just basic staining of all cells in the brain, the cerebellum back here, and here's the forebrain. In an animal who sings for a 30-minute period, we found that you get this whopping increase of mRNA expression in these song nuclei in the forebrain that I showed you in the previous diagram that I labeled red and yellow. When an animal is hearing playbacks of song, you don't see activation in these song production areas, but you do get activation, and now we know what is the auditory cortex equivalent of the songbird brain. And when a songbird is deaf, that auditory induced expression goes away, or most of it goes away, and he still sings. So in other words, a deafened bird singing, even if it's singing deteriorated deafened song, shows activation in these brain areas that are involved in the production and learning of the song. It's less than what you see in hearing intact animals, and that's because these deafened animals tend to sing less. That is, the amount of gene expression produced in this half-hour period is correlated with the amount of song produced. And then we made an accidental discovery that also would be informative for the mouse experiments recently, and that is we find that, yes, there are these activated regions due to singing, and there are exactly 7 of them, as I mentioned, in the songbirds and the parrots and the hummingbirds. Well, we found exactly 7 brain regions surrounding those song nuclei that are active in the production of movement behaviors. In this case, an animal hopping in a rotating wheel for the first time. And we found that, and that's shown in white here, you can see that the gene activation is directly surrounding those song nuclei. And the neural connectivity of these areas around the song nuclei is similar to the neural connectivity of the song nuclei themselves, forming in this case what we call a cortical basal ganglia thalamic loop. I know it's a full loaded term, but it's a common term used in neuroscience that's involved in motor learning and projections out of the forebrain that control motor neurons. So this led to the hypothesis, one more slide that's going to lead to this hypothesis. We found that if you were to play sounds to a non-vocal learning species like a ringdove, you see the hearing-induced gene activation like we see in the vocal learning birds. In other words, they do have the auditory pathway. When they perform movement behaviors, in this case, I couldn't get ringdoves to hop. They like to walk, so I had to walk on a rat treadmill. All right? And what you see is, it looks like one large area, but it really is 1, 2, 3 areas stacked up on top of each other, 2 here and another 2 lateral in the brain, 7 areas where you would expect to find song nuclei, but without holes of expression where song nuclei are located. So this led to this hypothesis that was the alternative to the intelligent design hypothesis that people were telling me about, and that is what I'm calling the motor theory of vocal learning origin, where I argue that all species have this vocal innate pathway, and what happened is that they also have a vocal, non-vocal motor pathway involving forebrain regions, basal ganglia structures, and that somehow during evolution, I argued that this pathway duplicated itself, formed new connections onto this innate pathway, and that duplicated pathway then basically replicated the motor learning pathway to now form an emergent vocal learning pathway. So this is the background that now we are working with, this hypothesis at least, with the mouse song system. And I thought the mouse was just going to have this pathway, and what we can do is try to influence new connections onto these neurons in the mouse brain, which it does have for production of innate sounds, to have a motor control pathway from the forebrain control the vocalizations. So, so to try to get back to the mouse now and ask what brain areas does it have that are maybe like or not like humans, what we did is we took, like what we did with the songbirds, we took mice and reduced them to sing in long bouts of what we're going to call mouse song. They like to sing to females, but we didn't want the females to act as a stimulus. So what we do is put female urine in their cage, and it's been shown that female urine has a potent pheromone that induces their singing behavior. And I showed you before this one little snippet of song, but that one little snippet comes from long sequences of this ultrasonic song that the bird sings. The sound is just condensed, so I can fit this 45-second of sound in there. I'm not going to play it for you, but it basically sounds similar to what I played before. And then have them produce this ultrasonic singing for about 30 minutes, as we did with songbirds, dissect the brain, do the in situ hybridization to measure the mRNA of these genes, scan the entire brain for vocalizing-driven gene expression, if it exists at all, and then compare to control groups like your silent, your deaf singing, and hearing-only controls. And this is the result that we obtained. We scanned throughout the entire forebrain and compared to an animal's hearing playbacks of sounds. We found one region in the cortex, one specific region here, including cingulate cortex, secondary and primary motor cortex above the level of the anterior commissure, that showed higher gene expression compared to a hearing-only animal, or an animal who was in silent control conditions. And the hearing-only animal didn't show difference. What was different here with songbirds is that even at baseline conditions, you do see some gene activation or expression in this area, as if there's something else that might be happening. And this is in the motor cortex. But nevertheless, quantitatively, it was clear that there was induction here shown in this graph. If you just focus on this part of the graph here, here is the singing animal who's hearing, relative expression to the silent control, and then the animal who is hearing this animal sing, okay? And then a deaf animal who's singing but can't hear. And it even shows a little bit higher expression in this case. And in the deaf animals, or let's say in the hearing intact animals, when they hear the playbacks of vocalizations, this is further back in the brain, you do get activation in the primary auditory cortex, that's shown here, as opposed to the M1 region. And when they're deaf, that activation goes down, or basically supporting the fact that we actually really did deafen these animals. So based upon this result, and there's a lot more to it, but I don't have time to get into all the details because I want to move on, the answer is yes, we did find cortical, and I just skipped that, and striatal regions right here in the dorsal striatum, right below this cortex region, that are active in the production of mouse ultrasonic songs in the absence of auditory feedback. And we know it's not pheromone stimulation because if we just, some mice who smell the urine don't sing, don't show this activation, or just olfactory stimulation. If you give them ethanol, you don't see it either. Okay, so what about connectivity? Well, for connectivity, here I'm showing you now sections through the songbird brain and the human brain of this pathway in the forebrain that's involved in song learning and this other pathway involved in the production of song. And a big deal has not only been made out of the presence of forebrain areas that control vocalizations and vocal learners compared to, say, a chicken, which doesn't have any of them, or a macaque, which has an area that, when stimulated, causes vocal fold movements through ambiguous, but doesn't make a direct projection to it. A big deal has been made out of this projection here from the forebrain synapsing directly onto the motor neurons that then control the muscles. This direct projection has, to date, only been found in vocal learning species, songbirds, parrots, hummingbirds, and humans. It's been looked for in the last 50 years by many laboratories and non-human primates and never found. It's been looked for in guinea pigs. We haven't found it in chickens or pigeons and several other bird species. So a number of scientists over the last 50 years, starting with Hans Kuipers in the 1950s, have hypothesized that this projection is perhaps one of the most critical for the evolution of vocal learning and spoken language. And Fisher and Hammerschmidt, it's Kurt Hammerschmidt and Julia Fisher, who actually was connected here at NIH recently, said the most important derived feature in the human lineage appears to be the evolution of the direct pathway from the motor cortex enabling volitional motor control over the vocal folds. And there's a reason why I brought this quote out, and you'll see in a minute. I just want to show you some of the evidence in birds and humans. If you place neural tracer into the RA, that motor output nucleus of songbirds, and go down to the vocal motor neurons in the brainstem, the white signal here basically is axons coming from the cortex. And you can see there's a heavy innervation of the motor neurons and respiratory premotor neurons here in the songbird that has not yet been found in a non-songbird. Human literature is not as robust because you can't do similar kinds of experiments in humans, but if you look at post-mortem stroke victims or trauma where there's been damage to the face motor cortex, and look at nucleus ambiguous motor neurons here in the human brain, you can stain for degenerating axons that projected there, and you can see these stripes here, many axons that are at least inside of nucleus ambiguous next to these motor neurons. In non-human primates, so far in the literature, the drawings have been shown, so I don't have the primate data to show you, but here's nucleus ambiguous with tracer injected into the motor cortex or premotor cortex of non-human primates, and they basically say no connections have been found. So what about mice? Well, I just assumed mice didn't have it, and other people actually wrote in their reviews mice don't have this projection, but no one's actually tested it. So we just tested it, and the way we did that is to inject transsynaptic tracer, pseudorabies virus, into laryngeal muscles, and these tracers, they have the property of they'll jump synapses, so they'll get taken up by the axon, infect the cell body here, and after 12 or so hours, then jump another synapse and go back, and if you get forebrain connectivity, they'll jump another. And depending on the timing of the tracer you see in the connected region will depend on whether if it's a first order, second order, or third order connected neuron. So we did that, and we found the white signal here is basically the tracer. We found, yes, after a day, we can find a tracer in nucleus ambiguous in the mouse brainstem, as expected. We can find it in the solitary nucleus, which we know from other work already projects to nucleus ambiguous and controls vocalization for respiratory linking with vocalizations. So that made sense. Second order connections we found later on, the next day, back into what was called the central gray, periaqueductal central gray, which is known for many years in mammals to control the production of innate vocalizations. So that makes sense, and this is the midbrain here. And then we went to the forebrain, and we scanned throughout the entire forebrain and didn't see anything except for one region. And that's shown right here, above the level of the anterior commissure in the exact location where we found the singing-driven gene expression in the mouse forebrain. And not all of the region showed backfill of the tracer, just the M1, the primary motor cortex region, as you would expect. And the neuronal morphology of this region here and its positioning in the cortex basically indicated it was layer 5 pyramidal neurons with their long dendrites going up into the cortex, which is what you expect if you're in the neuroscience field. You know that these are the neurons that send long projections out to the spinal cord and the brainstem. So this was a surprise for us to find this. And then we thought, well, maybe the connection is there and people have missed it before. It's just indirect. and somehow our transsynaptic tracer jumped synapses pretty quickly. We only saw this whenever we got the perioccipital gray backfill. So it suggests it was a direct projection, but doesn't prove it. So to test that, what we did is try to verify the projection by injecting another tracer called biodextranamine into the M1 motor cortex. And this tracer doesn't jump synapses, and we're going to see where does this M1 region project to. And we want to see if it projects to the motor neurons here. To make sure we identify those motor neurons accurately, we also injected cholarotoxin, a different tracer, labeled it in brown, that will then fill up the motor neurons here and won't jump in synapse. So we're asking, do these two meet? And so here's some of the data. We injected a lot of tracer, that's why you see some damage here, but we injected a lot of tracer because we want to fill as much of the motor cortex as possible, for, at least the singing activated part of the motor cortex. And we found that this region projects to, these are axons here, to the part of the striatum that showed the singing-driven activation. So that made sense. And then going down to the brainstem, here is your cholarotoxin, backfill from the motor neurons that project to the muscle. And here are your axons coming down from the cortex. And we look to see if they met there. And the answer is yes. This is shown high power now, and we found many examples of this. This is a motor neuron that synapses onto the laryngeal muscles, and these black lines here are axons that came down from the primary motor cortex. And some of them make multiple contacts, and you roughly find 1 to 2 axons per motor neuron, or 2 to 3 contacts. And so this demonstrated that the direction is the projection is direct. But there is a difference of what we had seen in songbirds and even the limited material in humans. The projection is pretty sparse. It's only 1 or 2 axons per motor neuron, whereas we can find tens, if not hundreds, coming down from the cortex in birds to the vocal motor neurons. But nevertheless, it made me wonder, did folks who had studied non-human primates miss this projection? Or are mice more similar to humans than primates are for this connectivity? And there are a lot of people who are doubting that mice would be more similar to humans than non-human primates, and I'm sure most of you would doubt that. So I mentioned before that those who did the work in non-human primates, particularly for the last 20 years, presented drawings, but not the actual primary data. So the last person who published on this was Christina Simonians, who actually worked here at IDCD, and for her PhD thesis before she came here, she actually tried to verify the absence of this projection in non-human primates, and I visited her lab, and we went through our mouse and primate brain sections together, side by side, to see if in macaques they don't have this projection from her cortical injections. And what you see here is the Nissl stain section in nucleus ambiguous and the reticular formation around it. And you can see many axons in the reticular formation next to nucleus ambiguous from her cortical injections, but we could not find any axons in the primary data here, not even this single one. So that suggests that there is a difference. But I will come to the end that there might be other explanations for these differences beyond species differences, but I just want you to hold on to that thought. Another thing we learned about is auditory connectivity. There's another hypothesis different from the direct projection from the cortex hypothesis. And that hypothesis goes that in humans, we have the auditory cortex sends a direct projection to premotor and maybe motor vocal areas that non-human primates do not have. That non-human primates may have a Broca's-like voice area or speech area, but they just don't have proper connectivity to these regions. And this region, this connection famous in neuroscience is called the arcuate fasciculus. So when we were looking at the connectivity of our mice with the injections in the motor cortex, it surprised us to find a distinctly labeled area compared to all other areas in the secondary auditory cortex, layer 3 neurons of the secondary auditory cortex, which are known to be the type of neurons that connect one cortical region to the other. And so we actually found such a projection, And we also found that this cortical region projects to a part of the thalamus, and that's what these black lines are, and the black dots are cells that project up to this motor cortex region. So it looks like it's forming an integrated circuit with the striatum, with the thalamus, and coincidentally with the auditory cortex, not visual cortex in this case, for this part of the striatum. And then just to show you in 3D diagram, here is the primary auditory cortex, secondary is next to it, it's sending a projection here. And we still need to verify it with the tracer injected here in the auditory cortex to show axons going up to the M1. So, to summarize this part of the experiment, what we have shown here is the mouse and songbird and the human and the chicken and the macaque brain again. And the mouse brain connectivity is looking more and more like a songbird and a human than it is a macaque or a chicken, with some differences. In this case, a weak or a sparse projection from the primary motor cortex as opposed to a heavy one in songbird and human. So it's not identical, but it looks like something more of degree rather than absolute difference. So the next question we ask is whether mice require the motor cortex to produce these vocalizations. We don't know if they're learned or not, but do they require it? In songbirds, whenever you knock out or basically lesion these motor pathway song nuclei, you eliminate the ability to produce song. It's not even a aphasia of song. You just eliminate the ability to produce it. Innate calls are intact. Now I'm going to show you an example here from my former PhD advisor's work. Here's a canary song. I'm going to play the sound for the video folks here. I can't quite follow. This is a canary song that's learned. Sounds a little different than mice. All right. And here is a canary that has HVC, lesion, and he's trying to produce song, opening up the beak, getting out faint little sounds, but actually no learned song. So they can do there's some innate alarm calls, and they can scream when you pick them up and so forth, but they don't produce this nice melodic learned vocalizations. So in humans, if the laryngeal motor cortex, or basically the general oral facial cortex region, is damaged, the region that makes that direct projection, we also lose the ability to produce speech. We can still cry, we can still laugh, we can still go ah, ah, and make what's supposedly innate sounds, but we can't produce learned speech. At least that's what I read in the literature, and that's what's been told. In mice, what do we know? We didn't know anything. So we actually placed lesions in the M1 motor cortex, as well as in the visual cortex for a control group, and performed sham surgeries for another control. And to do this experiment, we had to actually find a way to categorize these mouse vocalizations because they were quite variable. And so we categorized them by their frequency profile, that is no pitch jumps. In this one, here's a pitch jump that goes down. That's what the red dot means. A pitch jump that goes up. And we were able to get roughly 8 to 12 different categories of vocalizations. And then we, in these animals that we performed the lesions, we recorded those vocalizations. And then afterwards, we injected the pseudorabies virus again in the laryngeal muscles. And now sham surgery treated animals, we verified that the layer 5 neurons are still there. In the other animals, the M1 motor cortex lesions, we can see very few neurons left. And this is just to show you the percentage of the layer 5 neurons that are gone in these animals. And then we looked at their songs, before and after lesions. And what we found is, yeah, sham surgery treated animals are just fine. They're singing songs before and after. But motor cortex lesion animals, unlike songbirds and unlike humans, were also singing what looked like a song afterwards. Not little faint peeps or sort of innate type of or simple sounds. But we also noticed something different. If you notice when I'm pointing to these syllables here, they're showing a lot more frequency modulation than what the mice had produced before the cortical lesion. And that's shown quantitatively here. The red bar is the motor cortex lesioned animals, And this represents basically the standard deviation of the pitch, and the FM means the frequency modulation. Both of them are much higher after the lesion than before compared to the sham surgery of the visual cortex lesions. And to show you that in a more diagrammatic, in a distribution, showing the frequency of the pitch and the percent of the syllable repertoire within that frequency range, you can see before the lesion, there's a more tighter distribution of the pitch, where after the lesion, this animal is producing pitch that's much more distributed, indicating less control or less modulation, less control of the modulation of the vocalization. And at the time we discovered this, a paper came out last year from a colleague of mine showing that when male mice, so this is males in this case, go from pup early vocalizations to adulthood, they have a wider distribution in their pitch that then becomes more narrowly focused as adults. And so what we think might be happening here is that the motor cortex may be having some control over the pitch or the frequency modulation of the vocalizations, whereas the brainstem may be controlling the actual production of the sounds, which is similar to something that is found in songbirds, not for learned song, but for learned calls, where a male songbird has innate calls, and it will learn to modify those innate vocalizations with cortical input for whatever reasons. And if you lesion that cortical input, afterwards the bird can't sing the learned song, but it can still produce the calls in their innate form. So that's the hypothesis we're working on that. So I'm going to put this as a partial yes, that they do require partial input, or there's partial requirement for the vocal motor cortex to produce the vocalizations, in this case to produce a more sharper tuning of those vocalizations. So what about the auditory feedback? In songbirds and in humans, as I mentioned earlier, when we become deaf, our vocalizations deteriorate. And here is an example from a colleague of mine, Alison Dupe and Michael Brainard, on the zebrafinch. So zebrafinch sounds different than the canary. I'm going to play this. Can we have that volume turned back up? Here you go. I'll play it again. Okay, it has an echo there, but you get the point. And here is roughly a month in a young animal after being deaf. Right. You can tell it sounds more varied. It sounds less, let's say, stereotyped. Whereas I don't have the recordings of this, but in a suboscine songbird, it's like the chimp of the songbird world, before and after deafening, they did not see any differences in the syllable structure in these simple phoebe songs. It's called the Eastern phoebe. So what about mice? So we deafened mice. It took us a little while to figure out how to deafen them, but to remove the cochlea. And I'm going to play you the song now pitched down to the human hearing range again, but slowed down. So this is before deafening of this animal. All right, you get the point. It almost sounds like a whale when you slow it down. And you can see, just without me even playing it yet, that roughly 8 months after deafening, it took time, but it did show up. You had to wait. It looked like there was some degradation in part of the time that the bird was singing these syllables here, particularly for the more complex syllable type. And this is what it sounds like. So you can hear the raspiness in that sound. So it's not it's degradation. It's not as robust as the type of degradation you see in humans and songbirds, but it's there, as opposed to not being there at all. And that's shown quantitatively here, the time course we took to see it. Roughly around 3 to 8 months, you start to see these robust differences. The frequency of the sound goes up, and also the standard deviation of the pitch. In other words, the sounds become more noisier. The red line is the deafened animals. And in humans and songbirds, when we're deaf early in life, our speech is even worse than later on. And so we had a hard time getting viable deafened mice early on as pups, because the ear canal is closed. It's hard to do the surgery. So what we did is went for a genetic tool, the caspase-3 knockout animal. Caspase-3 is a gene involved in cell death. Those animals show normal motor behavior, but they lose hearing within the first few weeks after being born. And listening to their songs, basically before deafening, it's similar, so I'm just going to play afterwards. I call it the Mouse Godzilla. So this is also more detrimental. Now, there could be secondary effects here that we don't know about. We have to test this in other animals who are also deaf with other genetic disorders to be certain about it. But in terms of genetic mutations, it's the most aberrant song that I've heard of in any mouse that someone has knocked out a particular gene in. And this is just to show you quantitatively that the pitch goes down, there's more noise in the spectral purity of these vocalizations. So you can find it quantitatively. The simple sinusoidal type syllables, there are more of them. That's the orange labeled area here. And just to show you, we can find, as shown in the original papers in these caspase knockout animals, that the ear hair cells are missing in these animals compared to the wild-type C57s. So I'm going to say, yes, a partial requirement on auditory feedback to both maintain and develop the vocalizations. The caspase-3 knockout animal is dramatic. The adult deafened animals are not as dramatic, and that's why I'm calling it partial. But nevertheless, it's there as opposed to completely absent. So what about vocal imitation? We had a hard time testing this because it's hard to quantify these mouse vocalizations. And not only that, we were seeing mice actually show changes in their vocalizations, but we couldn't figure out what was causing it. And we noticed that these two different strains that we measured, the caspase-3 animals, or their wild-type precursors, and another set of mice called BXDs, were producing their songs at different pitches if you kept them in same colony conditions. And we mixed them together, we saw the pitch start to change, but we couldn't get a reliable change. Then we discovered that if we took a C57 male, pair it with a BXD male, and a BXD female, what happened is that those C57 males change over the course of 2 to 8 weeks. Well, really by 8 weeks, the pitch of their songs changed to come down to the level of the BXD male that they were housed with. And we thought it was the female that was could have been driving that change. So we did the complementary experiment, it's C57 male, a BXD male, and a C57 female to match that male there. And again, the C57 males went down. The BXD males went up a little bit this time. These are box plots. So they're representing the full range of the pitch. So you can see there's no overlap in any of the animals before cross-housing, and now you have much more overlap by the end of this 8-week period. So is this pitch imitation? Different animals, you can see the range here. There is a range, roughly a 20 kilohertz range in the pitch distribution even of the BXDs before you house them, and likewise a range here. So we looked at the difference of the pitch of individual cage mates and graphed that difference and found that those individual cage mates, even though their ranges were different, they were converging onto the same pitch range to each other over this 8-week period. And just to show you this, in a more dramatic form, before and after, here is the pitch difference before the cross-housing condition, and here is the pitch difference afterwards. Some animals went to basically a 0 pitch difference in their songs. And there was one male pair where at 6 weeks they were at 0 pitch difference, all right? And then the following week, one of the animals shot way up to go way out of the range. When my interpretation, if I'm going to put a human bent to this, is stop singing in my pitch range. Otherwise, I'm going to change or you change. So this is evidence of pitch matching. I don't want to call it pitch imitation yet, but it's as close as we get to some form of imitation. We didn't see changes in the repertoire composition, but the repertoire composition didn't differ between these two strains to begin with. While we were working on this study, a colleague of mine, Kazuo Okanoya and his group, were also trying a similar experiment in another group of mice where they cross-fostered pups of two different strains at a young age and claimed to not see the fostered animals shift up in pitch with their foster parents, claiming that the vocalizations are innate. But what they did in their experiment is they only cross-fostered them for a 3-week period. When we cross-house for 3 weeks, we also don't see the change. It has to be anywhere from like 4 to 8 weeks. And you really need to wait that full 8 weeks to see that change. So they're repeating their experiment based upon our findings to see if we can explain this difference between studies. So we get, yes, here again, I don't know how far vocal imitation goes in these mice, but we do at least have pitch matching. All right, so now I'm going to end off with genes and then summarize. So are there molecular similarities between songbirds and humans that we can ask for the presence or absence in mice? And we didn't know an answer to that until the last few years, and even more so recently. But there have been hypotheses that the HVC nucleus is similar to Broca's area in humans, that the striatum would be similar to part of the striatum in humans, that when damaged leads to aphasia deficits, and that RA, which makes that direct projection, is going to be functionally analogous to the laryngeal motor cortex, which also makes the direct projection. And to test this hypothesis, we did laser dissection of the song nuclei in songbirds and the surrounding areas here, pop them up to an Eppendorf cap, and then isolate RNA, hybridize into microarrays. And the same thing was done by the Allen Brain Institute with human brain. It's a much larger brain, of course. They dissected 900 brain regions from roughly, from 2 different people, a man and a woman, post-mortem, of course, and isolated RNA and also hybridized them to microarrays. And fortunately, they make this data publicly available, which they did this past year. And we compared our data with the human data. And to do this, to compare 900 brain regions with these song nuclei in songbirds, we asked, is there any gene expression difference in the song nuclei of songbirds, and we also did parrots and hummingbirds, compared to the surrounding brain areas that say this is a specialized gene expression pattern that you find in the songbird vocal region? Can you find a similar specialized pattern out of these 900 dissected locations in the human brain? And the answer, we might not find anything. And the answer was yes, and we demonstrate that in tree form here. So this is part of the frontal cortex, and these are different regions of the frontal cortex. And for the RA song nucleus in songbirds, there was one region, I highlight in green here, that had a strongly significant gene expression correlation with the precentral gyrus. The central sulcus part of the precentral gyrus is that's at the ventral bank of the cortex, where you find face motor cortex. And that was quite exciting, and it was highly significant, as I said. A number of these genes that showed the specialized expression showed evidence of, if anybody's heard about this, positive selection on their coding sequence as well in zebrafinch relative to chicken, and we also now see in humans relative to non-human primate. And looking at these patterns, what we find is that the RA gene expression profile is most similar to laryngeal motor cortex in humans. HVC, we don't find any parallel, at least not yet. And area X, it is the head of the caudate putamen in humans compared to all other striatal regions. And to show you an example of some of these genes is what's called a heat map. 3 different individual zebrafinches, 3 individual budgeriggers, which are a parrot. Blue means down-regulated relative to the surrounding cortical areas. Red means up-regulated. And we find a number of axon guidance genes in this list. The top one being SLIT1, which happens to be a target of FOXP2, for those who have heard about that gene. And the human FOXP2, gene mutated, which causes speech deficits, regulates SLIT1 more robustly than the chimpanzee version of FOXP2, and we can see it down-regulated in this motor cortex region. So we studied that further, and we verified that the FOXP2 gene, this is now what's called the archopallium, it's the neurons that project out of the forebrain. The psalm nucleus part of the archopallium has down-regulation of that FOXP2 gene in the songbird, the hummingbird, and in the parrot, but not in the ringdove or the quail, which are non-vocal learning species. So we asked, well, what about mice? And so we looked at the motor cortex region where we found the singing-driven gene expression in mice. Let's go here. Sorry. Whoops. Here's this motor cortex region. Here's our singing-driven gene expression in songbirds, in mice. We found some genes, which we just published on, like not only the axon guidance molecules, but calcium binding proteins, which are overexpressed in these song nuclei of songbirds. And we did not find overexpression in the motor cortex region relative to other cortical regions in mice. So that it wasn't like a vocal learner in this case. The same thing with the FOXP2 targets, SLIT1. Here's the downregulation again compared to the other neurons that project out of the forebrain, whereas in mice, it's not, overall in layer 5, it's not lower than the other adjacent cortical regions. ROBO1, it's ligand, the receptor for SLIT1. It has also a differential pattern, isolated cells with expression in RA. We don't see such a differential pattern in the motor cortex. In fact, it's a little higher. In this case, it is a little bit closer to the songbird situation. Then a paper was published this year. We started to think the mice are not like songbirds in their gene expression profiles. But a paper published by Hitchcock et al. In Neuroscience had shown that in the mouse forebrain, there are isolated layer 5 neurons, sparsely distributed, that express FOXP2 in the red signal here, and CTIP2, which is a molecule that tags cells that make projections out of the forebrain. So like in songbirds, just not as many, we have these layer 5 neurons expressing some of these genes, but just not at high levels. And we're going to ask the same question, is SLIT1 expressing these neurons more sparsely distributed? And I'm going to end off with a FOXP2 story just to tell you what we know about so far in songbirds and a little bit of information we got in mice. I'm going to play to you a sound file of children at different developmental stages that have a FOXP2 mutation and show you how it affects speech production. Could you turn the sound up for this? Whoops. Okay, here we go. All right.
Your name? Where do you live, Laura?
So this is a 2-year-old child.
And
it's hard to understand what she's saying.
Where do you live? And how old are you?
They are sure.
So this is someone who's older.
It's different. They are 6.
And he's 6 years old.
Their name is Tommy. That is Howard. That Howard was 6.
Okay.
And
I'm going to stop here. That's someone who's 11. Basically, by the time they get adult, they get better and so forth, but they're not good at speech production in the way that a normal child is. They're better at comprehension, but even comprehension is affected, but not as much as speech production. In songbirds, I'm going to go quickly here so we can finish up. In songbirds, what we find is FOXP2 is expressed in the basal ganglia as it is in humans and other vertebrates. It's upregulated in a juvenile bird during the critical period for vocal learning. And when a juvenile now becomes an adult, it's downregulated in the area song learning nucleus. So there's a correlation there. And my colleague Constance Scharf, after we published that study, what they did is injected an RNAi hooked up to a GFP molecule here, a green fluorescent protein, in the area X nucleus showed that it actually downregulates the FOXP2 here, where it's expressed. And shown here, the protein is downregulated, but not other genes like actin. And then give these FOXP2 knockdown animals a tutor and have a control group that has control hairpins that also has a tutor. So I'm just going to play you the tutor song. I'll do that again. Here is the control knockdown. It's a pretty good imitation. Here is another tutor. And here is the FOXP2 knockdown 2T. So you can hear the difference. Like humans, the birds can still sing, okay? Like humans, they can still learn something about the song. You can see there's some resemblance in the syllables here, but they don't accurately learn the syllables, the structure of the syllables, and they get the sequencing incorrectly. So this is a parallel. What about mice? Well, there's been a number of studies done on mice, but a lot of the behavioral work, I would say, has not been up to the kind of par that we've actually applied to songbirds in humans. And so we started collaborating with Simon Fisher, who's well known for his studies on FOXP2. They generated knock-in mice with the KE family mutation. And some people have studied those mice and found that they vocalize less. But that's all they've looked at. They haven't characterized much further. We began characterizing those mice, and what we found is that you have to do a heterozygous, because if you do homozygous, on both chromosomes, the animals die. So humans, only heterozygous humans survive. So this is a mouse with the KE family mutation, and what you can see just by looking at these two sonograms, I hope you would notice that at least this example, the sounds look more simple. And if you do a plot of the repertoire here, you see there's more simple sounds in their repertoire. And you can see that here also the frequency variance, that is the frequency modulation, is much less in the FOXP2 knock-in, KE knock-in mice, whereas the actual pitch at which they sing is no different. So it's not affecting all things about the vocalizations, but a lot of the modulation. And so this is similar, or maybe even opposite of what you might find in children, depending on your interpretation. But it is affecting the vocalizations here. So, so I'm going to say that we don't find gene expression specializations in mice brain that we find in humans and songbirds, but we do find a partial requirement on genes like FOXP2 as we find in songbirds and in humans. So I'm going to say none yet for the gene expression specializations. So what is going on here? I think that, that vocal learning may not be a dichotomous trait. Speech, who has it, who doesn't have it, that ability may not be dichotomous. That mice are, I'm going to propose, are limited vocal learners and somewhere intermediate in this continuum that I propose that may exist. And that mice, if that is the case, if you believe me, mice serve as good genetic models for some properties thought to be unique to humans for speech-language disorders. And given that it could be convergent, I would say, or let's say if continuous, you might be able to use mice to try to genetically enhance the circuit. And this is a hypothesis I'm going to be testing next, or I'm trying to test in my lab, is now we have these molecules that we know are robustly differentially expressed in the human brain circuit for speech, and in the songbird and the parrot brain circuit for vocal imitation, and that you don't see in the mice. And what we're going to try to do is to overexpress some of these genes or underexpress them, depending on the pattern in humans and songbirds, in the mouse M1 region to see if we can actually enhance this projection that already exists. So the mice, this story has now made this, this goal of mine easier because the mice already has a connection. 1 to 2 axons per motor neuron. Can I make it 10 axons per motor neuron and allow the mouse to potentially have greater volitional control over its vocalizations? And I will give credit to the funders for this particular project, the NIH Director's Pioneer Award, who allowed me to go in this risky direction, Howard Hughes Medical Institute as well, and NIDCD are one. And I have to give credit, this is my team here, although I mentioned this is the work of mostly 2 people in the lab with collaborative work from others, and I'll end there. Thank you.
time for a few questions for Dr. Jarvis. I'd like to remind everyone there's a reception in the library immediately afterwards with refreshments provided or sponsored by FAES. So.
All right. Yes. Oh, Dan.
Yeah,
Dennis.
hi. So that is a lot of very lovely data. So as I think you know, another group has deafened newborn mice on day one using
Yes.
a different mechanism. And at the moment, I think they're claiming they see normal development of mouse songs. So what do you
I wasn't
make
sure
of
if I could
this?
share those data because it was unpublished, but they did mention it at a meeting, yes.
Yeah, so what do you think?
Another group has, so there have been several groups looking at this. They have a different strain of mice. Well, there's a way of feeding these mice a drug that causes their ear hair cells to die off at an early age. And they start losing the ear hair cells about 2 days old, and they're gone at about 10 days. And mice don't really start hearing until about 12 days. And they claim to see no effect on the vocalizations on the mice, and they've looked at many different parameters. And I don't know the answer as to why the difference is there, except to say that I know that they didn't look at the frequency modulation, which is what we looked at, and the pitch distribution. The pitch of what I call that, the frequency range of the pitch. And with that group, we decided we're going to switch data sets and analyze the data with each other's approaches to see if we can actually reconcile what the differences are. Yeah. Yes?
Hi. I wondered if anybody had recorded the vocalizations of wild field mice in different neighborhoods, and if it were distributedly different in different neighborhoods, if that would be informative.
Yeah, that's an interesting question because before I even started down this mouse path, it was already known that counterintuitively, vocal learning bird species in captivity sing more complex songs than their wild-type counterparts. And that is not because we're selecting, it looks like the females are selecting for this. The more varied the sounds, the more the female likes it. And so what's selecting against it? The idea is predation, or at least my hypothesis is that you don't habituate to varied sounds that easily, even predators. And so, so I was thinking maybe what's happened is that Jackson Laboratories unintentionally are evolving vocal learning in the laboratory with these ultrasonic courtship songs, and maybe laboratory mice are more down this path than wild type. And if that were true, then the wild type mice should sing more stereotyped songs. And someone published a paper last year saying that wild type mice do sing more stereotyped songs. So what we'd like to do now is to get wild mice and actually do the tracer injections to see if they actually do have this projection or not. Yeah? Okay.
Well, I guess that's it. Thank you for your attendance.
Thank you. All right.